Hello everyone, my name is Kelly Gonzalez, and today I will be going over my Virginia Sea lesson plan titled Where in the Water is the Atlantic Sturgeon? First, a little bit about me. I am a master's student at the Virginia Institute of Marine Science, and I am interested in Atlantic sturgeon population success and habitat preference. This lesson plan is based on a portion of that work, and I'm very excited to share it with you. So the focus of this lesson plan is to investigate where Atlantic sturgeon are found in those upper tributaries of the Chesapeake Bay, while also exploring abiotic stressors and how those influence habitat preference and use. The Virginia Standards of Learning that will be focused on with this lesson plan are Environmental Science 1, where the student will demonstrate an understanding of scientific and engineering practices, and also Environmental 5, where the student will investigate and understand that the Earth is one interconnected system through which energy and matter flow. You will get a list of vocabulary words that I encourage you to give to your students before the presentation, as they are included in it and also in the worksheet and handouts. And there will also be a presentation to go over with your students. This will include many open answer questions that you can give time for students to answer out loud or just maybe ponder on. But the presentation will go through some background information. So, to talk about why we're looking for Atlantic sturgeon, there are some leading questions for students. The first is, if fishing has been reduced, what else could be holding them back? This is where we get into some abiotic factors. The second leading question you can give to your students is, what is the best way to find sturgeon? Given that they are endangered, making them rare, and a highly migratory species. A potential answer to that leading question is the use of environmental DNA, and this is where we get into our methods for this project. So you can give them the real-life eDNA steps that I do in my lab. For the in-classroom activity, groups of 4 to 5 students will receive 8 paper cups labeled 1 through 8, containing a mixture of colored and white clear beads that they will have to pour over a filter. That filter will be placed in a plastic strainer and be held over a bowl. Now, you can use water with this activity, or you can choose not to, but just keep in mind that water best represents what I do in my lab. You can also use gloves and forceps for this activity, but it's completely optional. It just further supports the idea that this is a very sterile procedure. Once they have poured their water over their filter, they will extract the clear white beads using those forceps or just their hands and count how many they get. And they will record that number on their data sheet along with the water quality measurements provided on a field data sheet that was taken by our researchers in the field. And thank you very much. I hope your students enjoy this lesson plan and learn a little bit more about Atlantic sturgeon and our Chesapeake Bay. Thank you.